Okay, so where we left off, we have our search for the DNA structure and molecule. Now remember the logic in Avery, McCarty, and McLeod. Very, very lovely, elegant experiment. We have our suspects, RNA, DNA, or protein. Those were the big things that we think could be the transformation factor. So keep in mind, that's what we're thinking is going to happen. We think that one of these chemicals is going to transform R cells to S cells. And just like the imposter, if we vote them out, so to speak, and they are indeed the imposter, then the killing should stop. In this case, the transformation won't happen. So let's take a look at what happened. And then I would ask you to think about what that means. So RNA digesting enzymes, we get rid of the RNA. So there should be no RNA. And then we add this to live R cells. And then we're going to just see if transformation happens. And sadly, the mice still die. So transformation is still occurring. Now, what I want you to think about then is if the mice still die, does that mean that RNA is the transformation, or does it mean that it's not the transformation factor? All right, so let's take a look at the protein digesting enzyme. We're going to remove protein, and we're going to see if protein, by removing protein, if transformation still occurs. And unfortunately, sadly, for our poor little mice, the mice still die. And so transformation still occurs for our little mouse when we remove protein. And in each of these cases, if I grow the bacteria that's found inside the mouse, I find S bacteria. Now, the last case, we have DNA digesting enzymes. And in DNA digesting enzymes, we remove DNA, there's no DNA, we add that mixture to S cells. We add that mixture to R cells, and lo and behold, transformation does not occur. The mouse remains healthy. And so not only that, but if we then look at the bacteria inside the mouse, we only find R bacteria. So transformation does not occur. So think about it. We remove DNA and transformation does not occur. So think about what that does. Fill out your boxes. Does this mean that RNA is the transformation factor or not? Does this mean that protein is the transformation or not? Does this mean DNA is the transformation factor or not? Okay, so hopefully you caught the fact that RNA is not the transformation factor. RNA, if you remove RNA, transformation still happens. If you remove protein, transformation still happens. We still find S cells. It's only when you remove DNA that transformation does not occur. And so very, very elegant experiment to kind of set up a step-by-step logical conclusion. Now, unbelievably, people still wanted to not accept DNA as the transformation genetic molecule. And so remember, that's what we're ultimately doing. What is the genetic molecule? Remember, everybody thought proteins. Now we're slowly chipping away at it. It doesn't seem that proteins are the genetic factor that carries traits. But we still were kind of stubborn. We still thought that proteins were more likely. So along another decade goes by. How do we know for certain? Alfred Hershey and Martha Chase again used a very, very elegant experiment using bacteriophages. Now, bacteriophages are very wacky looking viruses. They infect bacteria. We don't really care about them we care about them as research, but they don't infect us. They infect the bacteria in us. And so bacteriophages, one thing that we found out about them that really made them pretty handy, they are made of only DNA on the inside and protein on the outside. Now, here's where you have to remember about proteins and nucleic acids. Proteins contain sulfur, but no phosphorus. DNA contains phosphorus, but no sulfur. And so Martha Chase and Alfred Hershey did a really, really clever thing. They used radioactive isotopes to label our viruses. And so they made two batches. One batch made viruses that were made with radioactive sulfur. Now, radioactive sulfur, if you grow viruses in a medium with, of course, the bacteria that they infect and radioactive sulfur, you're going to get bacteriophages with radioactive protein coats. Now, the DNA is not. That's real important to catch. The DNA is not radioactive. So that's one batch. We're going to make bacteriophages with radioactive protein. Then we're going to make a separate batch that's grown in radioactive phosphorus, and we're going to make radioactive DNA. So, wonder of wonders, now we have a way to track both protein and DNA. It would be like me wondering where do you guys go after AP Biology? And so maybe I spray some of you with bright pink neon paint on your backpacks, and then I spray bright blue spray paint on some other backpacks, and then I just watch for where those backpacks go. So remember, the question is, what is the genetic material? And in this case, we're trying to figure out what goes into the bacteria, because what goes into the bacteria is what's ultimately infecting it and causing it problems. And that's our genetic material. So they did a very, very, again, very nice, elegant experiment using the time-honored equipment of a blender. So we have our two batches. Remember, one of them is going to be protein. One of them is going to have labeled DNA. And literally what we're going to do is watch where the radioactivity goes. So in the first batch, the bacteriophages infect with DNA that is not labeled. The DNA is not labeled, but the protein is. And so what they did is they put all of the bacteria, the solution, into the blender, and they just spin the bacteria around to shake off the bacteriophages. And then we're going to centrifuge, and the centrifuge, remember, spins around, and that's going to pull the cells down at the bottom because they're really heavy, or heavier. So they're going to be being pulled down to the bottom, and then the liquid is at the top. So keep in mind, where is the cell and where is the bacteriophage? We have shaken off the bacteriophage, so they're going to be in the liquid. And this time we're seeing radioactive bacteriophages, and so they're going to be in the liquid. And that's where the radioactivity is. And then the cells, which do not have any radioactivity, are going to be down at the bottom. And so they could just measure, where do I see the radioactivity? They're going to see the radioactivity in the liquid, but not in the cells. And then the proteins are, what their conclusion then was, that the proteins are not going into the cell, it's the DNA. The next batch, we're going to look at the radioactive DNA. And again, same thing happening. The bacteriophages are injecting the DNA into the cell. This time that means the radioactivity is inside the bacteria. So when we shake them off with the blender, the bacteriophages are still going to be in the liquid, but now the liquid is not radioactive because the proteins are not the ones that are labeled. The DNA is. And so when we spin this down, we're going to see the radioactivity in the cells because that's where the DNA is. So the conclusion, radioactive DNA, is at the bottom in the cells, and that the conclusion was the DNA is going into the cells. And so Hershey and Chase was kind of the they kind of had the experiment that put the nail in the coffin of the idea that proteins were the genetic material. And that's kind of where we're at with our experiment. And so what we're going to do in the first half of class on Friday, we will watch the next step in DNA history. So, all right, that is our introduction. Those are our background materials, and happy studying.